In this tutorial, we're going to talk about how to use histogram data and guide the simulation as the external force in MELD. And the central idea is illustrated in the following figure here. And we'll make use of unpredicted protein residue pairwise distance histogram data as an example. So in the beginning, we have to understand what does histogram data mean. In this example, we have the residue pairwise distance histogram. So for each histogram, it basically means the probability of residue pairwise distance along a certain distance range, say from 2 angstroms to 22 angstroms. As you can see for this red histogram, this residue pairwise distance is most likely to be in this range because the probability is much higher than the rest of the distance range. So our first step is to apply this filtering to figure out what are good histogram that we can make use of in the simulation. And then after figuring out what are the histogram that we can make use of in our simulation, we can convert them into the restraint energy, and we'll talk about this step later. As we talked before, we should have a distance histogram data file, and for each histogram, first evaluate where the sum of probability over a certain range, and we have 5 angstrom here, is larger than a certain threshold, say 0.8, which indicates where the most possible distance range between these two residues. After this step, for processing all the distance histograms, we will obtain a contact file for the pairwise residue distances. For example, in the first line indicates the predicted lower bound distance between residue 1 and residue 13 is 14.3 angstroms, and the upper bound will be this value plus the filtering length we used, which is 5 angstroms. So the upper bound of this residue distance will be 14.3 plus 5 angstroms. And we'll make use of these two numbers as restraints in our simulation. Then we can start to set up our system in MELD. First, we import some required modules, and we have to make sure that we have the sequence file available, and we will build our system starting from the sequence file that contains the sequence we want to fold from a linear chain. And we will select a force field for this system. In this protein folding simulation, we usually use this force field. And the other thing is we have to select a temperature range along all replicas. We'll make use of the geometric temperature scalar available in MELD, which basically says from the lowest replica to the 40% of all replicas, the temperature ranges from 400 Kelvin to 550 Kelvin. And from 40% to the highest replica, it will maintain as the highest temperature, which is 550 Kelvin. This whole script will set up the basic environment for our system. And then we will integrate this process histogram data in MELD. So after the first step, we will have this contact file, and we'll make use of this contact file now. So the basic idea is that for each process histogram, we'll convert it into a flat bottom harmonic restraint, where the restraint between lower bound and upper bound is set to be 0, and we'll have restraint energy penalty outside this range. This can be clearly seen in this picture, where we have restraints equals to 0 within this range and have energy penalty outside this range, which basically means that we want to guide these two residues to this distance range that they're supposed to be. In addition to these distance restraints, and we have scripts for converting predicted dihedrals into restraints in MELD simulation. You can find the full sample setup scripts here and make some modification for your own use. So after having these setup scripts, you can start to generate the starting point of our simulation, which are the data directory and the log files. After having these two directories, then we can start to run our replica exchange simulation on our queuing system using the corresponding command. Thank you for watching. If you have any questions, please post your issues on our MELD GitHub page.